And this is our final module in the course on cell communication. And this video is on general ideas into chemical signaling. So I just want to give a brief introduction on the different ways that cells can communicate with each other through chemical signaling. And then we'll go from there into a little more detail in the coming videos. The image I've shown here is a scanning electromicrograph of the cell body of a postsynaptic neuron. So you can see this sort of globular structure here. That's the cell body. And these structures that are stuck to it are the synaptic terminals of what are called presynaptic neurons. So this is actually a chemical synapse inside the nervous system. This is a fixed specimen that was prepared for electron microscopy, as we talked about in the early part of the course. So it doesn't look exactly like this in life, but most of the details are there. And so we're going to talk about, for example, ways in which other neurons can communicate with cells in the nervous system and in other systems using chemical communication. And that involves talking about things like neurotransmitters and other chemicals that are released from cells, as well as receptors that lead to transduction, signal transduction. This is a word that I used earlier in the course, and I'll come back to those ideas in these videos and describe them in more detail. So I'm going to talk first about 4 different types of signaling that occur between cells, and that involve chemicals. One is contact dependent, in which this is the first model here in A in the figure below. This involves a signaling molecule that remains bound to the cell surface. And we've looked at this already in terms of the immune response. So we talked about, back to the lectures on apoptosis, we looked at the Fas ligand and the Fas death receptor. This was a form of communication where cells directly had to make contact with each other. So the Fas ligand, for example, was a transmembrane protein, and the receptor was also a transmembrane protein of another cell. And those two cells came together and allowed the ligand to bind to the receptor. So this is contact-dependent signaling. So this can happen in development, where developmental cues are required for cells to proliferate or divide, and as we've seen in the immune response or in apoptosis more specifically. Then there's paracrine signaling. If you haven't heard of the word paracrine, this is your introduction to it. It sounds a little bit like endocrine, which is another type of signaling, but paracrine is more general in the sense that basically it's just a cell that releases some sort of factor, which is here indicated by a local mediator. So you might be mediating things like proliferation again, or division of cells, or other signaling mechanisms that don't require contact. So basically a cell is releasing some sort of chemical factor, and it's being taken up, or not taken up, it's binding to receptors of other target cells, and therefore initiating some sort of cellular signaling mechanism. And so paracrine basically means within the vicinity of a cell. Okay, so this is a secretion of a signaling molecule, and importantly, this involves diffusion of short distances with local effects. Okay, so paracrine signaling is not something that takes place over a very long distance, or very great lengths in tissue. It's something that is important over very short distances, like cell widths apart. So local cells that are not connected by contact dependent signaling or any synaptic contact, but undergo paracrine signaling. And then there is synaptic signaling. We're going to talk about synaptic signaling in more detail. And I alluded to that with the title slide, with that electromicrograph showing presynaptic and postsynaptic neurons making contact. In this case, signaling molecules are delivered from target cells directly, from the cell of origin to a target cell, directly across a chemical synapse or a gap. And this is specific to neurons, or cells of the nervous system. So here's a neuron, here's a cell body, here's the axon. We've looked at neurons briefly so far in the course. This is the nerve terminal, where neurotransmitters are stored in vesicles, and here they're being released from the synapse. So the synapse is a space between the presynaptic and the postsynaptic cell, so pre, before, post, after. And those chemicals bind to these transmembrane receptors and initiate signaling, intracellular signaling mechanisms, and we'll look at those in more detail. Okay. And then there is the endocrine signaling mechanism, and I think you've probably heard of endocrine signaling in the endocrine system. This is different from paracrine, because paracrine, as I mentioned, is signaling across short distances in local tissues, local cells. Endocrine signaling is a type of signaling that takes place over a longer distance within an organism. So an endocrine cell will secrete a hormone, for example, that will be taken up by the bloodstream and delivered to target cells. It's a slower process. It involves the bloodstream, but very important biologically because there are a number of different endocrine factors, hormones that can be released from different parts of the body and have effects on other parts. Now I'd like to say a word or two about concentration. You might be wondering, well, what sort of concentrations of chemicals do we find in these different forms of signaling? Well, they can be quite variable, dependent upon cell types and chemicals, and so I won't give any real details, but I'll say generally, when we look at endocrine signaling is a type of signaling that is in which we find target cells are very sensitive to endocrine factors or chemicals. And working concentrations, or endogenous concentrations, I should say, in these cases, are something on the order of nanomolar. 10 nanomoles per liter, for example, might be what we would find for a hormone to its release at a higher concentration, but by the time it finally gets to a target cell, it's been diluted, it's traveled such a long distance. And for that reason, target cells can be very sensitive to very small concentrations of hormones in the endocrine system. With synaptic signaling, for example, or paracrine signaling, we're looking typically at higher concentrations. These might be in the micromolar range. So endocrine signaling typically is a process whereby target cells and their receptors are highly sensitive to these diffusing factors. Now, once we get to a target cell, now we have to start thinking about receptors. What's going to be mediating the transduction of this signal? So cells communicate with each other via chemicals. I've mentioned this already. And this signal can take on different forms. When a cell is a cell that's going to initiate a signal, that signal starts off as a cellular response. There might be something happening inside the cell. It might lead to a depolarization. That might be an electrical change in the membrane. That might then release or lead to the release of a chemical. Now we have what was originally a cellular signal, some sort of intracellular change, then leads to ultimately a release of a chemical. That's sort of the final output of that cell, pushing or giving a signal that is meant for another cell. What happens next is that chemical is transferred to another cell, there's binding, and then we have a conformational change of a receptor. So now we're looking at we go from a chemical signal, now we have a conformational signal, so this is more of a biochemical protein change that's occurring. And that's going to now be transformed again into an intracellular signal. So that signal went from being intracellular to being extracellular to being intracellular again. And that's how a signal can be transferred from one cell to another. And so it's important and informative to talk about how that signal is transformed in the target cell, how it gets from outside the cell, from being just a chemical that's released into the extracellular space, how it gets transformed into now a signal that the cell can use, and how that can lead to some downstream effect on gene expression, for example, or physiological change. So this is signal transduction, a term I've used already. And we're going to look at three major classes of receptor protein complexes that transform these extracellular binding events into an intracellular signal again. So something is outside the cell, it has information, it's a chemical, As soon as it binds to a receptor, it's now going to be transformed into something else. And we'll talk about that. Three different types of receptors. One we looked at already, ion channel linked receptors. Oh, sorry, no, it's the number 3 we've looked at already. In these videos, we'll look at number 1, ion channel linked receptors, in which an extracellular signal, which is a ligand, binds to a receptor. These receptors are also ion channels. So it's one complex. It's a receptor, so it's one part receptor, it's one part ion channel. So as soon as something binds to it, it opens the channel, and ions flow down their electrochemical gradients. And that is important information in terms of the cell. And examples of that will be glutamate receptors and acetylcholine receptors that I'll talk about in the next video. After that, in another video, I'll talk about G-protein coupled receptors. These are what we call metabotropic receptors. So the first one are ionotropic receptors, because their activation leads to a flow of ions. And G-protein coupled receptors are metabotropic because it's some sort of, well, the term metabolic, so it leads to some sort of, I wouldn't necessarily call it metabolic, but the idea is there in the sense that it's an intracellular signaling mechanism. That's why it gets this prefix metabo. So it's a type of receptor that isn't itself an ion channel, but it initiates a signaling mechanism that will ultimately lead to some change in the cell, activate some sort of pathway. And enzyme-linked receptors, these are the ones that we've talked about already. I'm not going to go into more detail there because you can, if you're interested, you can go back to those lectures. But this is when we looked at growth factor binding. So you recall nerve growth factor and how it brought these enzyme-linked receptors, which are two large subunits brought together. There was autophosphorylation, and that allowed for the broadcasting of different signaling pathways or recruitment of other proteins that led to the phosphorylation of downstream partners. And these are just those 3 different groups illustrated by cartoons. So here's the ionotropic receptors. There's a little ion binding domain that allows for opening of the channel. So, for example, glutamate might bind here. The signaling molecule in this case looks the same as ions. That's not actually the case, but it's highly simplified. I'll show better schematics in the next video. But as glutamate or acetylcholine can bind with these regions of the receptor, open it up, and allow for ions to move through. G-protein coupled receptors, here's the GPCR here, the G-protein coupled receptor. And then there are G-protein, there's the trimeric G-protein complex that we'll talk about. That there's a very specific pathway there that leads to activation of downstream proteins. And these are the enzyme-coupled receptors that we've already covered in the course. So more general information. Again, I'm going to talk about these ionotropic receptors in more detail in the next video. But basically, they're composed of several subunits with multiple transmembrane segments, just like any other transporter channel that we've seen. These tend to be localized at very specific sites within the plasma membrane, more specifically localized to a synapse. So if we think about where neurotransmitters are important, I think we would all conclude that they're important at points at which cells communicate with each other. So in the case of neurons, we have a cell body, a long axon, and then a nerve terminal. The long axon, we find mostly sodium channels along an axon. And we looked at sodium channels in a previous lecture and how they're important in action potential propagation. But the cell body and the terminal, that's where a lot of the communication happens. If you imagine, one neuron and another one and another one, so you have a chain of neurons communicating with each other, or perhaps you can imagine in a circuit, a network of neurons with cell bodies and nerve terminals. The terminals are where the neurotransmitters are released, and the cell bodies are where the postsynaptic receptors are found. So this is where we would find ionotropic receptors at synapses, because this is where a neurotransmitter is released and where they need to act. They reversibly bind neurotransmitters. So for example, acetylcholine might come in, bind reversibly with a receptor, allow for opening, and then they dissociate. it would be pretty useless if binding was permanent. If we had receptors that were on a postsynaptic membrane, the acetylcholine would be released, bind, and then we would have sort of a very long period where the receptors are open, allowing ions to move through, and we have no recovery. We would have, in the case of acetylcholine and muscle contraction, we'd have muscle contraction but no relaxation. And so what would happen over a longer period of time would be the receptors would have to be endocytosed and replaced with receptors that weren't bound with ligands. So to alleviate that problem, chemicals reversibly bind with these ionotropic receptors so as to initiate a signaling pathway and then dissociate, and then that signal would then shut down, and we'd have back to a resting state. So channels open, ions move down their electrochemical gradients, as we've seen in other examples. And importantly, as I'll talk about in the next video, what happens is when these channels open, we have an increase in membrane permeability to a specific ion, or an increase in conductance. So basically, these are electrical words or words in biophysics that we use to describe the movement of ions across a membrane. So we can say that there's increase in permeability, so there's now a pathway for ions to move. There could be electrical conductance because ions carry a charge. And if that's happening, there's going to be a change in excitability. And I'll talk about excitability in the upcoming lecture, but just to put this out there so you've been exposed to it one time already, and we'll talk about it the second time in the next video. But excitability is basically a tendency for a cell to, like a nerve cell, to respond in the form of an action potential. So cells have a resting membrane potential that is usually around minus 70 millivolts. But in order to initiate an action potential, as we've seen in one of the previous lectures where I talked about, there was an experiment that we did, sort of a thought experiment that I covered in one of these slides, where current was injected into a membrane, and we looked at sodium channels, and there was a square pulse injection of current to depolarize a cell, and there was a little increase that was associated with that. It's I think the last slide in the sodium channel lecture. That little increase elevated the membrane potential a little bit to surpass this threshold level. And once a nerve cell gets past this threshold, it goes on its own and produces an action potential. Because what happens is that's enough, that little lift is enough to activate sodium channels that are voltage dependent. Okay, so that little injection of current, that's a positive charge that lifts the membrane potential past this threshold. Now an action potential is formed. So if a cell is gaining a positive charge, we're making it more excitable. So I can inject current, positive current into a cell. This is giving it a positive charge. And I'm making it more excitable. We can do the same thing. We can open up an ionotropic receptor, and we can allow positive ions to get in. And that is also making it more excitable. Okay. So giving a cell more positive charge increases its excitability. And I'll talk about that some more. So neurotransmitters are very important because they're chemicals that are produced in the cytosol of a neuron and are stored in the presynaptic nerve terminal. And there are a number of biosynthetic enzymes that are important for these reactions in producing neurotransmitters. And they're delivered from the cell body to the nerve terminal by slow axoplasmic flow. The neurotransmitters themselves are biogenically produced at the terminal of the cell, the nerve cell, and are stored in synaptic vesicles. Some neurotransmitters, we call these neuropeptides, are actually sorted through the ER and the Golgi, as are their proteins, and delivered by fast exonal transport. But by and large, what I'm going to talk about are neurotransmitters, and I'm really only going to use two examples, glutamate and acetylcholine. And these are both excitatory neurotransmitters. One, glutamate is important in the central nervous system. Acetylcholine is very important in the peripheral nervous system for muscle contraction. And these are the ones that we'll talk about primarily in the course. So signaling at a chemical synapse, just as a refresher for some, and I've mentioned this already, that this idea of a chemical synapse, but here it is in sort of schematic form. Here we have a presynaptic terminal of a neuron. Here are synaptic vesicles with some sort of neurotransmitter inside. Here's the postsynaptic cell. This is the synaptic cleft, and these are receptors at a presynaptic terminal or postsynaptic terminal. Okay, so the synapse is basically a specialized region between two adjacent cells. I've put here that they're electrically isolated just to let you know that one cell is not attached to the other in any sort of electrical way. There are actually cases where cells are not electrically isolated. I certainly won't talk about those here because that would, I think, unnecessarily complicate the discussion, but these are two separate cells that communicate only via chemicals between them. Okay, and so this is a resting cell, and this is a cell that's active. The cell membrane has been depolarized. Vesicles are fusing with the presynaptic membrane. You recall fusion of the membrane, release of contents into the extracellular space, and binding of the ligand to the sites on the receptor. And this allows for opening of these ionotropic receptors. Here is a transmission electromicrograph showing what a real synapse looks like. And so I think in many cases in cell biology, the cartoon is, I think, more informative. That's kind of the case here too, unless you know what you're looking at. If you can understand this, I'd hope that you can understand this, because this is a transmission electromicrograph, and we looked at some TEM images earlier in the course. And of course, the whole idea of that was so that you would be able to understand what these images represent and what they mean. Of course, this is just a slice, a very thin slice, only nanometers thick, through a synapse, which we have. There is a postsynaptic cell here in blue, so this is the postsynaptic cell, and these are two presynaptic terminals. So you can imagine that there's one cell, that's the target cell, and there are two presynaptic cells kind of on either side. But again, this is a slice through it, so we're really just seeing one snapshot, one little slice of this entire mechanism here. And you can see all sorts of little vesicles that are part of what we call the readily releasable pool of vesicles that all contain neurotransmitters. Some are lined up here getting ready to fuse with the presynaptic membrane. Same with the other presynaptic cell. And here's the postsynaptic cell. There's a mitochondrion right there. What you find also is what's called the synaptic density. There's a little thickening of the membrane here. It's also indicated. I don't think it's not labeled in this diagram here, but it's indicated in the little cartoon here. The synaptic density is basically, the pre and postsynaptic densities are basically regions of membrane that contain a greater density of protein complexes in the plasma membrane. So they're integral membrane proteins and other membrane-associated proteins that are important for constructing a synapse. In some cases, they might be higher expression of receptors, possibly even cells that are important for communicating with the extracellular matrix and other cell types as well. Anyway, this is what synapses look like in cells. We have pre- and postsynaptic cells. That the presynaptic cells are where these vesicles originate, and they fuse with the membrane, and those chemicals are released on postsynaptic receptors, which are located within these synaptic densities. And this comes back to this idea of, where do we find ionotropic receptors? We're more likely to find them at regions where there are synapses between different cell types. Now, there are different types of chemical synapses, and I'll define these as either excitatory or inhibitory. I've already talked about excitatory synapses. So in the case of glutamate and acetylcholine, when those receptors open, there's an increase in positive charge that we then can measure inside the cell. That's because sodium is coming in and carrying a positive charge with it. That makes the cell more excitable. So this increase in excitability which is its ability to depolarize a cell. That's what we call an excitatory synapse. Okay? A synapse where if it's activated, receptors are activated, that is going to cause an increase in excitability of the cell, and most likely that's going to be an increase in positive charge inside the cell. There are also inhibitory synapses in which when these receptors are activated, there is a decrease in excitability. So an example here are the alpha aminobutyric acid receptors, the gamma aminobutyric acid receptors, or GABA receptors. Now these are the most common inhibitory type receptors that are found at synapses in the central nervous system. Activation of GABA receptors leads to a decrease in excitability because typically what happens is there's an influx of chloride ions. So instead of sodium ions coming in, it's going to allow chloride ions to come in. So there's a different selectivity here, and rather than letting positively charged sodiums to come in, negatively charged chloride ions are going to enter the cell. So that brings in a negative charge and reduces the excitability of the cell. So it has the opposite effect. So that's how we define an inhibitory synapse. Now, for those of you that are studying cell physiology or perhaps neurobiology, this forms the basis of networks in the nervous system. So now we can have excitatory synapses and we can have inhibitory synapses. And you put that all together in a network. You can imagine a network of maybe 5 or 10 neurons that are all connected or communicating with each other. Some have excitatory synapses, some have inhibitory synapses. Now you have a way of really driving a network of information in a collection of cells. And you can expand that to a greater degree, and of course you get a nervous system or a brain, which is essentially a very, very complicated network of many different pathways of excitatory and inhibitory synapses. But we won't talk about much of that in this course. Really just want you to understand at the cellular level what the characteristics are of excitatory and inhibitory synapses. Okay, and for this video I'll leave it there, and I'll come back with more information on different receptor types and how we, more specifically, how we can define these excitatory synapses.